What if we could cure disease by editing our DNA like a Word document? Thanks to a breakthrough called CRISPR, that future might be closer than you think. Let's break it down simply. Inside your cells is DNA, the instruction manual for building and running your body. It's written in a 4-letter code, A, T, C, and G, and sometimes there's a typo. Just one wrong letter can lead to serious problems. For example, with sickle cell disease, a single letter change makes red blood cells sickle-shaped, blocking blood flow and causing pain. With cystic fibrosis, a mutation creates thick mucus that clogs the lungs. With hereditary blindness, faulty genes prevent light-detecting cells from working properly. These are just a few of the thousands of conditions rooted in our genetic code. For decades, scientists dreamed of fixing these errors, but gene editing used to be slow, expensive, and imprecise. Early tools like zinc finger nucleases and talons could make changes, but they were clunky, hard to design, and not very precise, like trying to fix a typo with a chainsaw. Then came CRISPR, and everything changed. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, a long name for a surprisingly elegant idea. Bacteria use CRISPR as a defense system. When attacked by viruses, they save a piece of the invader's DNA and use an enzyme called Cas9 to cut that sequence if it shows up again. Scientists realized this could be used to edit genes in any organism. With CRISPR-Cas9, we design a guide RNA to find a specific sequence in the DNA. Cas9 follows that guide and makes a cut. The cell then tries to repair it, and we can use that repair process to fix a mutation, disable a gene, or insert new DNA. It's precise. It's programmable, and it's fast. What once took years and millions of dollars can now be done in weeks for a few $100. CRISPR is already changing lives. Take sickle cell disease. Doctors remove stem cells from a patient's bone marrow, edit them in a lab to flip a genetic switch, and then reintroduce those edited cells. The patient starts producing healthy red blood cells, and in some cases, symptoms disappear. For hereditary blindness, CRISPR is delivered directly into the eye using a harmless virus as a carrier. Once inside the retina, it targets the faulty gene and removes it, allowing some vision to be restored. How you deliver CRISPR depends on the disease. Sometimes it's edited outside the body then reinfused. Other times, it's delivered directly to the target tissue using viral vectors or lipid nanoparticles, microscopic bubbles that sneak into cells carrying the CRISPR payload. But the potential goes far beyond medicine. In agriculture, CRISPR is helping create crops that are drought-resistant, tomatoes that don't bruise, wheat that resists disease, rice that grows better in tough climates, soybeans with healthier oils. And because these edits are made to the plant's own genes, without adding anything foreign, many CRISPR-edited crops avoid the GMO label entirely. In bioengineering, scientists are using CRISPR to edit pigs to make their organs more compatible for human transplants, program bacteria to produce medicines, fuels, and biodegradable plastics, create lab-grown meat that mimics real muscle tissue, build living sensors that detect toxins, pollutants, or pathogens, and the list keeps growing. And further, artificial intelligence is helping accelerate all of it. AI models can now scan the entire genome to predict the best place to make an edit and flag spots where off-target cuts could happen. That means safer, more accurate treatments. Machine learning is also helping design better guide RNAs, the pieces that tell CRISPR where to go, and simulate how cells might respond before anything is done in the lab. In agriculture and biotech, AI is speeding up discovery, optimizing cell factories, and even helping design proteins that can make CRISPR work more efficiently. Put simply, CRISPR gives us the tools to edit DNA. AI helps figure out where, how, and why to use them. But with great power comes big questions. There are two broad categories of editing: therapeutic and enhancement. Therapeutic editing means fixing or preventing disease. This is where most scientists agree CRISPR can do the most good. Enhancement editing means editing traits that aren't medically necessary, like muscle mass, eye color, or intelligence. That's where things get murky. Even more controversial is germline editing, changing DNA in sperm, eggs, or embryos. Those changes get passed down to future generations. In 2018, a Chinese researcher shocked the world by editing the embryos of twin girls in secret. His goal was to make them resistant to HIV. The global scientific community condemned the act, citing huge risks and unanswered ethical questions. He was later imprisoned. Most scientists agree, editing disease in adults is one thing. Editing the human germline is something entirely different, with consequences that could ripple for generations. And even beyond the ethical debates, the science of gene editing still faces major challenges. First, off-target effects. CRISPR is incredibly precise, but not perfect. Sometimes it cuts the wrong section of DNA, which could accidentally disrupt a healthy gene or trigger unintended side effects. That's why researchers are working on ways to improve targeting, and why safety testing takes time. Second, unpredictable repairs. Even when CRISPR cuts the right spot, the cell's natural repair process can be messy. It might fix the break in a way that creates new mutations, deletes too much DNA, or fails to restore the gene's function. Scientists are exploring better ways to guide the repair, but for now it's not always reliable. Third, delivery. Getting CRISPR into the right cells at the right time in the right amount without triggering the immune system is still one of the biggest technical hurdles. Some treatments involve editing cells outside the body and reinfusing them. Others require packaging CRISPR into viral carriers or tiny nanoparticles, but those delivery methods don't always reach the right tissues, and they can be inconsistent. And finally, complexity. Not every disease comes down to a single typo. Conditions like heart disease, Alzheimer's, or diabetes are influenced by dozens, sometimes hundreds, of genes plus lifestyle, environment, and other factors. Editing just one gene often won't be enough. In these cases, CRISPR might be just one tool in a much larger toolkit. So while the potential is enormous, the path forward is still full of scientific puzzles. That's why researchers are already working on next-generation tools. Upgrades to CRISPR that could make gene editing safer, more accurate, and more versatile. One of those tools is called base editing. Instead of cutting the DNA, base editors work more like a pencil, gently swapping out one DNA letter for another. It's perfect for fixing single-letter mutations, which cause a large number of genetic diseases, and because it doesn't break the DNA strand, there's less risk of unwanted repairs. Another breakthrough is prime editing. Think of it as a search and replace function for the genome. Prime editors can precisely rewrite longer stretches of DNA, inserting, deleting, or replacing sequences with fewer errors and more control. It opens the door to treating a much wider range of conditions. Together with AI, these upgrades may take gene editing from a miracle tool to a routine part of medicine. The future of gene editing is just beginning. We're still learning how to deliver CRISPR more safely, how to reduce errors, and how to treat complex conditions that involve dozens of genes, not just one. But the pace is accelerating. New tools like base and prime editing are expanding what's possible. Artificial intelligence is helping us find the right targets and avoid the wrong ones. And clinical trials are turning once theoretical breakthroughs into real-world treatments. Meanwhile, researchers, regulators, and ethicists are working to answer the bigger questions. Where should we draw the line? Who gets access? And how do we make sure this technology helps people without harming future generations? Gene editing isn't just about science. It's about choice. And it's redefining what it means to treat, prevent, and potentially design the future of life. That's all for this episode. Thanks for tuning in to Brain Snacks. Subscribe for more big ideas explained simply.